Hi students, let us pass on to the next part of the lesson, the structure of chromosome. Let's see some historical aspect of chromosome. The chromosomes were first reported by Strasburger in 1875 and the name chromosome was given, that is the term chromosome was given by Olayer in 1888. C.V. Bridges in 1910 described that the chromosomes are the carriers of the physical units called the genes, and it was T.H. Morgan in 1993 reported that the chromosomes are the carriers of hereditary information. For that he was awarded the Nobel Prize in 1993. Now, let us go into the structure. If we are taking a chromosome, the chromosomes are the carriers of the genetic material, having the information, that is the hereditary information or carrying the hereditary information. Now, the chromosomes, when present in the nucleus, they are highly coiled and condensed structures called chromatin. So, the chromosomes are made up of chromatin. The chromatin is packed with DNA, the one which contains the genetic material. So, the chromosomes are highly coiled, condensed chromatin fibers which are packed with the DNA. The DNA is the genetic material carrying the information. So, normally in a chromosome we have the genes. The genes are located on a chromosome at a particular point, and that point is the locus of that gene. What is locus? The gene is located at a particular point in a chromosome. That point is the locus of that gene. Suppose we are taking the gene capital R, and the capital R gene has a point or a locus. It is located in the fifth locus. So, locus is a point where a particular gene is located on a chromosome. Now, loci is the plural form. Loci is the plural form. Now, what is a gene? A gene is nothing but a segment of DNA. Segment of DNA. A gene is nothing but a segment of DNA carrying the information, and that information is inherited to the next generation. So, a gene is nothing but a segment of DNA carrying the genetic information which is being transferred to the next generation. So, during actually cell division, the genes which contain the hereditary information are passed on from one generation to another generation. I will go deep the structure of a chromosome. If you are taking a chromosome, particularly at prophase, a chromosome is a thin, long, thread-like structure formed of actually two symmetrical units, two similar units called chromatids. Now this is chromatid. This is called chromatid. So two similar or symmetrical units are chromatid. Now the chromatid, suppose you are taking a chromosome, it contains two arms. The arm which is shorter, now the two chromatids are joined together by what is called centromere. The two chromatids are joined together by centromere. Now one of the arm is shorter and one of the arm is longer. Now the shorter arm is called as the P arm. This is what is called short arm. The longer arm, this is what is called Q arm. The longer arm is called Q arm. So, formed of two symmetrical units called chromatids. The two units are joined together at a point what is called centromere. The longer arm of the chromosome is called what is known as Q arm, and the short arm is otherwise called as a P arm. Then we will go further. Now the chromatid is formed of highly coiled thread-like structures inside. These coiled thread-like structures are called chromonema. Chromonema, singular, chromonema. Now if you are taking the chromonema, if you are uncoiling it, this is a chromonema. At certain regions, the chromatins are accumulated and condensed together to form bead-like structures. These bead-like structures present in the chromonema are called chromomeres. Chromomeres. So this is a chromonema. The chromonema has bead-like structures all along its entire length, and these structures are called chromomeres. This is nothing but the accumulation of chromatin regions. Now, the chromonema is nothing but actually DNA. So the chromatid or chromonema, actually you see that one, chromonema is equal to, chromonema is nothing but chromatin fibers. The chromatin fibers are made up of DNA, proteins, RNA, and some metallic ions. So, the term chromonema and chromatin are similar. They are made up of linear chain of DNA with proteins along with RNA and then other metallic ions. Then, what are the other components? Now, the two arms are connected by means of a narrow region. That narrow region is called centromere. Centromere. So in a chromosome, we have constricted regions. The constricted regions are classified into two types. One, primary constrictions, another one, secondary constrictions. So the narrow regions present in the chromosome are of two types. One, primary constriction, another one, secondary constriction. The primary constriction is present in all the chromosomes. The secondary constrictions may or may not be present. The number of secondary constrictions may be one or more than one. The primary constriction is otherwise called as centromere. Or we can say the primary constriction consists of two components. One, centromere, another one, kinetochore. The two components of primary constriction, centromere, another one, kinetochore. Now this is the primary constriction. This is the primary constriction, otherwise called centromere, which also has what is called kinetochore. This is called kinetochore. What is kinetochore? Kinetochore is nothing but complex protein fibers to which the spindle fibers are attached at the time of cell division. Complex protein fibers to which the spindle fibers are attached at the time of cell division. So this is about the primary constriction. We also have one or more secondary constrictions. Now this is secondary constriction. Secondary constriction. So we have here I represented two secondary constrictions. Two secondary constrictions. I represent primary constriction, now secondary constriction. Now these secondary constrictions contain a gene for ribosomal RNA. The secondary constrictions contain gene for rRNA. So it is a region of synthesis of rRNA or ribosomal RNA. Also, the secondary constriction is responsible for the formation or organization of nucleolus. Hence, it is also called nucleolar organizer. Nucleolar organizer. So inside the nucleus, one or more spherical bodies. They are called nucleolus. These nucleoli are produced by the secondary constriction. Hence, they are called nucleolar organisms. They also have the gene for ribosomal RNA. So it is a place of synthesizing ribosomal RNA also. So that's about the primary and secondary constrictions. Now, in some cases, then one spherical knob-like structure is present connected to the main chromosome by a secondary constriction. A knob-like structure connected to the main part of the chromosome through an elongated secondary constriction. This knob-like structure, the round-shaped structure, and that structure is called satellite. Chromosomes having satellite are called SAT chromosomes. SAT chromosomes. So the satellite is not present in all the chromosomes. It is present only in specific chromosomes. For example, 1, 3, 10, 16 and Y chromosomes in humans. They have only satellite. So the satellite chromosome is used to identify the morphology of the chromosomes. So the knob-like structure connected to the primary chromosomes, the main part of the chromosome, through an elongated constriction, that is a secondary constriction, is called satellite. The chromosomes with the satellite are called SAT chromosomes, and they are used to identify the morphology of the chromosomes. Then, the terminal end of the chromosome is called telomere. The terminal end of the chromosome is called telomere. Now this is the telomere. So normally the chromosome has a polarity, just like north and south poles. Now what is the function of telomere? Normally it has a specific sequence of nucleotides, for example TTAGGG. This is the nucleotide sequence present in the case of telomere. Telomere, terminal part of the chromosome, having specific sequence of nucleotides TTAGGG. What is its function? It prevents the attachment of the chromosome to the adjacent chromosomes. It maintains stability. It provides stability to the chromosomes so that one chromosome may not actually attach itself to another chromosome. This is because of the presence of telomere. Now during cell division, during each cell division, the size of the telomere is decreasing. When the size of the telomere is decreasing, ultimately what is happening? It causes the age of the cells, and also it causes cancer. So it is responsible for the growth and the reproductive ability of the cells. That's about the telomere. I mentioned about the chemical composition. So it is formed of chromatin. The chromatin is a gelatinous material having DNA, RNA, proteins. What are the types of proteins present in what we call the chromonema or in the chromosomes? There are two types of proteins. One, histone proteins. Histone proteins. Another one, non-histone proteins. These are the two types of proteins present along with DNA, RNA and metallic ions. So there is a chemical nature of what is called the chromosome. The histones are responsible for providing stability to the chromosomes. So these are the fundamental structures what we have in the chromosomes. Next one we see the classification of chromosomes based on the position of centromere. Accordingly, there are 4 different types of chromosomes. Number 1, telocentrome. Here the centromere is present on the proximal end. The centromere is present on the proximal end. So we have only one arm and the chromosome is rod shaped. Now the second one, acrocentrome. Here the centromere is present at the one end of the chromosome. So that we have two arms, one short arm, another one a long arm. So the chromosome is rod shaped. And now the third one, the centromere is present just near the centromere. So we have unequal arms, one long arm, one short arm. So it gives the appearance of a J shape or L shape. So these chromosomes are called J shaped or L shaped chromosomes. Now the last one, the centromere is present in the center. The centromere is present in the center. So I am taking that one in the centromere or in the center. I am taking this way. And that is the two arms equal in size. Hence the chromosome is V-shaped. So either the chromosomes are rod-shaped or J-shaped or L-shaped or V-shaped according to the location of the centromere. Accordingly, we have telocentric on the proximal end centromere, acrocentric at the one end of the chromosome, the centromere, and the third one submetacentric. It is present near the centromere, what is called actually the centromere. And in the case of metacentric, now the centromere is present in the center, so that the two arms are equal in size. The chromosomes are classified into two types based on the functions. One type of chromosomes called autosomes. Another one, sex chromosomes. Sex chromosomes. Now the autosomes, also called somatic chromosomes, they carry the genes for the expression of the body characters. For example, in human being, we have 44 autosomes. 44 autosomes. The remaining two only sex chromosomes, XX in female, what is studied already, XY in male. So 44 plus 2, 44 plus 2. So the autosomes carry the genes for the somatic characters or the body characters. Now the sex chromosomes, they are also called allosomes. Also called allosomes, as they are different in nature. As we have two different types of sex chromosomes, they are called heterosomes. They are called heterosomes. The sex chromosomes are of two types, one X, another one Y. In the case of females, we have two X chromosomes along with 44 autosomes. In the case of male, we have one X chromosome and one Y chromosome along with 44 autosomes. Totally we have 46 chromosomes. Now the sex chromosomes carry the genes which are responsible for the determination of the sex, whereas autosomes carry the genes for the expression of the body characters, either the skin color or any other color, or even the yellow, the tongue rolling. These are all the external characters and they are being expressed by the autosomal genes. Students, I would like to pass on to the next part in that lesson, karyotype. What is karyotype? Before that, we have to know something about the chromosomes. In all the organisms, including human beings, we have chromosomes. The chromosomes are found in pairs. In human beings, there are about 23 pairs of chromosomes. 23 pairs of chromosomes. Out of these 23 pairs of chromosomes, 22 pairs, what is seen already, autosomes. Autosomes. The remaining two, what we have actually one pair. This is a sex chromosome, what is seen already. So this is one. That's what's the meaning of actually chromosomes. Then, in all the diploid organisms, sexually reproducing organisms, the chromosomes are found in pairs. The chromosomes are found in diploid condition. The chromosomes are found in diploid condition. That is, in the somatic cells, we have diploid number of chromosomes. During the gamete formation, either the sperm or the egg, the diploid number of chromosomes is reduced to haploid. So, in gametes, we have haploid number of chromosomes. Haploid number. This is because of the reduction in number of chromosomes during the formation of gametes, either the sperm or egg. Now I will come to the point. What is a karyotype? So it refers to the number, size and shape of the chromosomes. Number, size and shape of the chromosomes. Number, size and shape of the chromosomes. In a cell nucleus, the number, size and shape of the chromosomes in a cell nucleus is called karyotype. Now we can represent the chromosomes in a diagrammatic manner. And that method is called, that method of representation is called ideogram. Ideogram. What is ideogram? The diagrammatic representation of the chromosomes or the diagrammatic representations of the karyotype of a species. In what way we can represent the chromosomes in a diagrammatic way? And for that we have to make what is called karyotyping. What is karyotyping? All the chromosomes in a cell are isolated and are represented in a diagram in pairs. Such a diagrammatic representation of chromosomes is called what is known as ideogram, a diagrammatic representation of a karyotype. Now in that karyotype, the chromosomes are arranged as homologous pairs in the order of decreasing length. What are the criteria used for making a karyotype? 1. Order of decreasing length. Order of decreasing in length. The chromosomes are arranged based on order of decreasing in length. The thickness of the chromosome. The thickness. The location of the centromere based on the location of centromere. Then the shape of the chromosome. The shape of the chromosomes, and then just representing the sex chromosome or X and Y chromosome at the end. So while making an ideogram, we have to actually represent the chromosomes in the order of decreasing in length, the thickness of the chromosomes, the location of centromere, the shape of the chromosomes, and X and Y chromosome, the sex chromosomes at the end. This is called actually ideogram, the diagrammatic representation of the chromosomes. I mentioned we have to draw the diagram in the order of decreasing in length. Now the first chromosome, this is the first pair of chromosomes. Now the last pair we have, this is the last pair. This is nothing but what is called the X and Y. X and Y. So this is the first pair, this is the last pair. That means the first chromosome is the longest one or the largest one. The smallest chromosome is Y. Even when compared to X, the Y chromosome is the smallest. So we are arranging the chromosome in the decreasing order of length in the diagram, what is called ideogram. First one is the longest one, the Y chromosome is the smallest one. And we have in between the order of length is decreasing. We have also medium-sized, short and shorter chromosomes. So this is the way we are representing what is called ideogram, and that is what we are having karyotype of human being, either for male or for female. So while representing the ideogram, what I mentioned already, the X and Y chromosomes are normally represented at what is called the end in the diagram. So we have 23 pairs of chromosomes. These 23 pairs of chromosomes are grouped under 7 headings or 7 groups, A to G. A to G, this is what we have. The chromosomes are arranged in 7 groups. We will study in 12th standard about the 7 groups A, B, C, D, E, F, G. So in each group a particular number of chromosomes normally placed. Now in the case of human karyotype, there is no telocentric chromosome. Telocentric chromosome is absent in the case of human karyotype. We have either metacentric or submetacentric or acrocentric, and there is no telocentric chromosome. For example, the first chromosome, the first pair is metacentric. The Y chromosome is acrocentric. In between we have either metacentric or submetacentric chromosomes. That's about the karyotype and also the ideogram. Thank you. The following image shows the ideogram of human male and human female.